Kenne ich eigentlich nicht.
Pero después que se han convertido en sí, no.
I don't know how this goes, like this? Yeah. Okay.
It's okay, Laura.
I will check, but I think it should be already.
Done? You ready? Okay, 9 a.m. Welcome everyone to this session. We will have two amazing presentations. Yes. Remember all the students that will present that you have 15 minutes. Okay, 2 minutes left. I will say hey, something like that. In the tribunal, I am Jason Borrell from the University of Oviedo, the Dr. Eva from the University of Oviedo, and Laurence from the University of the Sorbonne. Enjoy. You have 15 minutes and the floor is yours.
Thank you so much, Ezra. My name is Laura, and I'm very excited to talk to you about spatial partitioning of large mesopredators on coral reefs. And a special thank you to my supervisors, Luciana, Conrad, and Mark. One thing, I think the camera should be on me for them to see me, right?
I have no idea.
Is the screen
And
Laurence?
I'm trying to check the.
Again, just to have a lot of people watching from Australia, so.
But I see you.
It's a camera.
Okay, perfect. So, welcome to Australia. Good afternoon and good morning, everyone here. So, I was at the Australian Institute of Marine Science in Perth, and on the right side here we see beautiful pictures from an artist from the Kimberleys, north of Western Australia, and it's called When the Freshwater People Meet the Saltwater People. And me, coming from Munich, a pretty landlocked area, suddenly researching the ocean, and before like only doing aquatic ecology, I thought like, yeah, that was very much showing my beautiful journey. So the first thing I want to do is I would like to acknowledge the traditional owners of the land on which I was researching and working in Western Australia. So I would also like to pay my respect to the elders, past, present, and emerging. And behind here we see a drawing of Makuru, which is the season of fertility, which is right now in June and July in Australia, in Perth area, and shows very beautiful the waterways. So what my title of my thesis already tells, talks about spatial partitioning. So what actually is spatial partitioning? To nail it down very easy, so individuals and species divide space to reduce competition and simply coexist. So if we draw a bit back and we think about our cohort, so basically I used the data of our cohort and modeled it with my code. So if we look at our cohort in October 2022, we see, okay, for our masses in general, we are pretty separated already. And we have a core use area depicted in red here of 1,500 square kilometers. And we have an extended use area here of 6,100 square kilometers, which is really big. But now looking at February 2024, we see everything extended incredibly. So our core use area is more than 12,000 square kilometers and an extended use area of 125,000 square kilometers. So what we have here is spatial partitioning. So basically we partitioned space according to our very interest, according to our niche, what we want to study. So we all separated because we can't all do the same. So if you would look now at us, all being here, being an analyzer of us, being all at the same place, you'd be like, why is everyone at the same place? So sometimes, of course, movement in space and time makes a lot of sense. Sometimes maybe from an outward perspective, not. So we keep this in mind. So the second thing was I was talking about large mesopredators. So maybe you ask, okay, what are large mesopredators? Everyone have heard probably of apex predators like tiger sharks, great white sharks, killer whales, and mid-ranking predators would be mesopredators. In my case, I'm looking at gray reef sharks and red bass. Gray reef sharks are much larger. They're around 1.8 meters, whereas red bass get around 70 centimeters. We have to say they are both very common mesopredators in coral reefs, and still there's a risk effect coming from the gray reef shark towards the red bass, where I have a little error here. And they're going for very similar prey. So for cephalopods, for crustaceans, for small fish, which is shown on the left. So we can consider them as mesopredators rather than predator and prey. And the interactions and the coexistence of mesopredators are really largely understudied in coral reef ecosystems. So how does this interaction look like? So we have the red bass swimming around here, sneaking a little fish very soon. And we have the shark going around. We have gray reef sharks and white tips. So this is the red bass. This was a gray reef shark. So as you probably noticed, like the gray reef shark was chasing a bit the red bass, but still it was not a predator-prey, typical predator-prey relationship. So we know now what is spatial partitioning. We know now what study species we have. Where did we do this study? So we see here two little frames. The upper frame shows a Scott reef, which consists of 3 atolls, of reef atolls, and in red we see Rolly Shoals, which is also 3 reef atolls. They are both around 400 kilometers off Australia at the continental shelf. So now we have something really interesting what we have to consider. So Australia again, in yellow depicted, we see the exclusive economic zone of Australia. Inside of the exclusive economic zone, we have this little box here, which is the allowance for Indonesia and for traditional fishers in Indonesia to catch sharks, to dry them, to salt them here, because this is just of the history of their what they ate or what they still eat. They still do. So, and now looking back to our study sites, so one of our study sites, Scott Reefs, is heavily fished and depleted of sharks in there, exactly where this little fishing box is. And Rolly Shoals, which is not fished and a marine protected area since the 1990s, it's a very prestige reef system, and there's a lot of sharks, a lot of abundance of sharks. So now we come to the research question. So we have gray reef sharks and red bass co-occurring in coral reefs. They both have a pretty similar diet. Research showed that already. So now we ask, do they partition in space? And this principle goes back to the very old principle of competitive exclusion and niche separation. So Lotka, Volterra, they were already talking about. So if you have species that have similar diets, they should somehow partition in space or in time. Otherwise, one would outcompete the other. So the research question is, do red bass and gray reef sharks exhibit spatial partitioning within coral reef habitats? And does that occur on smaller scales and on a temporal basis, if not on larger scales? So we predict that it occurs, and if not on larger scales, at least we can find it on smaller scales. Another thing that we really wanted to look at is we have, as I already introduced to you, many sharks at Rolly Shoals. We have fewer sharks at Scott Reef. So thus spatial partitioning changes with different shark abundances. So we predict red bass exhibits different space use according to the shark abundance. So how did we do this? We're coming to acoustic telemetry. On the left, we see the tagging procedure. So hook and line caught fish and sharks. They were surgically implanted little acoustic telemetry tags. They were seared and they were again left in the waters. And here on the right, we see a little scheme of the acoustic telemetry receivers. So they're on the seafloor deployed. And when a fish or a shark comes into this receiver area, it's getting detected. And yeah, that means, of course, if fish and sharks are deciding not to swim around in our receiver area, we have no clue where they are, what they're doing, where they're going. So now we look at our tagging locations. Here on the left, we see where sharks and fish were tagged. So of course, we tagged them somewhere where our receiver area is to make sure, okay, at least we have a high probability of them actually living there. So we see them inside the lagoon, outside, along the reef crest, and we see like a really extensive receiver area at Raleigh Shoals, which is the unfished habitat. Then we have Scott reefs. We also see the tagging was done here, and we see a slightly smaller receiver area here, also inside the lagoon, outside along the reef crest and in a channel. So looking the very first time at the data, what kind of data do I have here? In general, this data is already distributed by the sites, Raleigh Shoals, Scott reef, and by the species. So red bass and sharks, here were two data sets, that's why. So, and we see in the x-axis, we see the timeline from 2007 to 2016. And on the y-axis, we see the individual tag IDs. So what is very obvious right away, so every little dot is basically a detection at one of the receivers in the array. And what is very obvious, some individuals were really consistent, detected throughout the whole tag lifespan. So we saw them, we detected them always. And some individuals are really sparsely detected. So there's a lot of like time in between, no detections, you have no clue what they're doing. It looks like there's also some pattern, maybe we have time to come back to this later, and also some absences. In general, how to describe this is also possible with a residency index. So we can see how resident actually are there from 0 to 1, how much of the time that they have been in the receiver area are they actually also detected. So we see high residency indexes here. Looking at the receivers, we see something very interesting already. So Scott reef, so the receivers are color-coded. At Scott reef, we have receiver 4, which is super famous for sharks and for fish. Whereas at Raleigh Shoals, we see, okay, the most famous receivers for red bass are definitely not the favorite receivers of sharks. Interesting. So we continue with the hourly detections. What we found is that we have a pretty steep increase of hourly detections for the red bass in the late evening, afternoon, evening. Whereas for the shark, it stays a bit more stable, but also it increases a bit, and this can be also related to feeding behavior, nocturnal feeding behavior. So now, we go past the raw detection data. Now we are going to spatial metrics. This is what I did in the beginning with our cohort data. So what we have here is a probability distribution of 95% of the red bass being here in Raleigh Shoals 3 square kilometers. Being at Scott reef, almost the same size. With a core use area of 50%, so the very core use area of almost 0.4, 0.4, pretty similar. So this is where the most time core use is spent. Looking at the sharks, we see at Raleigh Shoals definitely a larger area, extended area, and also a larger core use area. So this is what we kind of expected. Then at Scott reef, we see that the extended use area is actually more like the fish extension area, and the core use area is slightly bigger. But we see here a trend, it's bigger at the unfished Raleigh Shoals. So now we want to answer the question that we asked in the beginning, spatial partitioning. How do we do this? So we overlap the kernel utilization distribution. So we did this with the 95%, with the extended ones, and we see, okay, we have a really great overlap in both sides. Of course, we assumed this because this is the receiver area, this is what we studied, this is where we tag them. But then looking at the 50%, we see the overlap here. There's barely overlap, it's only 18.6%, whereas in Scott reef we have 64%. So we have spatial partitioning here. So we have to go back to the results of the size distribution to understand this a tiny bit better. So what we see here, we have slightly larger individuals in Raleigh Shoals of red bass and of gray reef sharks. And there's unpublished data who shows that sharks at Raleigh Shoals are more abundant and are larger. So we're coming to the main finding. So at Raleigh Shoals, which are non-fished, we have many larger sharks. We found spatial partitioning. At Scott reefs, which is heavily fished, we have fewer and smaller sharks. And we found no spatial partitioning. So we were thinking, maybe here, there's not really like, red bass are not really scared or afraid of the sharks, whereas here, they really distribute their niches. What else did we find? So we saw in evening, afternoon, nights, elevated activity. We saw exactly the core use areas inside, outside the lagoon, along the channels. We saw the residency indexes. So what we can say is that unfished, pristine reefs, they show spatial partitioning. And we saw this on a small scale. And that suggests that we potentially, by fishing, we disrupt spatial partitioning. And we disrupt also trophic relationships within these coral reefs. So when fishing, always the larger individuals of like a trophic chain. So, and also all these findings, they show that MPAs mitigate actually spatial partitioning. So they maintain this. They maintain spatial partitioning. So overall, summing this up in 2 words, I would like to say, yeah, protect diversity. And I want to thank IMOS, AIMS, and the IMBRC, my references. And yeah, do you have any questions for me?
Thank you so much, Laura. We're on time, so. Any questions from the audience?
Yes.
Short question, right?
Yeah, it's that I have no idea about the ecology of sharks or the fish, but I do about the other ones. And I'm curious about, is there an ecological meaning for the 95 and the 50? Or?
So what do you mean? Like, so they have like both of them, sharks and fish, they use like a very limited space. It's like we do too. Like normally you don't travel every day in the world. So of course they have like their use spots and their core use areas. So they have some areas where they're like most of the time and where they spend like a lot of their times. And then there's extended areas where they also be. So it's like habitat.
Yeah.
Where they are.
I want to say first, thanks for the presentation. I really liked it. And second, I wanted to know a bit more about the tagging and how many did you tag, or yeah, what can you tell me about it?
I think I have it somewhere. So yeah, we have individuals at Rowley Shoals, Red Bass 15, at Scott Reef 10. We have a really extensive data set for the grey reef sharks, 35 for Scott Reef 11. So in total, it was a data set of more than 700,000 detections. And yeah, the detections per receivers are here, and also the receiver number, so, and the detections in total. Yeah.
Okay, so Lawrence?
Yes. First, thank you for your presentation and super nice images. I was wondering about the tagging. When you tag the animal, you can also record the things about if their sex distribution, for instance, or did you do that? And is there any importance, or I don't know if you have looked for some pattern or not?
That's a very good question. Thank you so much. So first of all, the tagging procedure was done way before I was studying this really data set, because that's long-term monitoring. So, but yeah, the sex was noted, but only for the sharks. And since I was looking at mesopredator interactions, it would have been really important for me to know also the sex of the fish, which we didn't have. So this is why, unfortunately, I couldn't include it in my analysis, but that would be very nice to do so.
Then I have another question also related to space partitioning. And the question is if you have some information on the occurrence of the preys at both sites, if the preys that are not, I don't know if they are fished by the by the people in the place where the, I don't know if the right to fish is only for shark or also for the prey fish.
The right is also for the prey fish, but actually they really target sharks there. So this is their main goal. So maybe what we can see.
I'm just wondering if you.
Yeah, that would be like actually to model or to look at the prey distribution would be very interesting. Maybe as you can see here, so they hang a lot around like the reef channels here, and we found also, or like a lot of papers state that actually they do this because to save energy and also because the prey goes basically through the channels and it's like they can just wait and be like, hey, I'm here. So this would be the next thing, also to look at the tidal flow and like environmental factors. So this would be the next stage, and then possibly would be very, very nice to have also some prey data. Get that. Thanks.
Okay, thanks.
Eva.
Thank you, yes. Well, thank you very much for your presentation. I really enjoyed it. With all the animations, excellent views of everything, with very nice pictures. It's really a pleasure to see this. See you have worked at it very much. So I'm very happy to see this, and also congratulations to your Horse Institute, because I'm sure they helped you a lot to get all this so good. Here, I'm like Jiminy the Cricket. This means that the first question is compulsory. Is I am sure you have, but have you the approval by a competent research ethics committee to tag the fish? I'm sure the answer is yes, but
You should mention.
Yes, yeah, it is. This is all approved, and many years ago already approved. Yes.
I'm sure.
Yeah.
But
Also in my thesis, it's already very like
good. But since it's compulsory, at least for Europe, and this is a European master, I'd encourage you, if you work with vertebrates or with any species that is included in the European legislation, that to have compulsory approvals by research ethics committees, it's good you state it since the beginning, so I don't pose you this question, and or even better, to write the reference, the code for the approval, which is nice. I mean, it's something quite routine, and it's good.
that's very good to know. Yeah.
Yeah, I know that sometimes it's difficult, but you just ask your supervisors, and you get this information easily.
Yeah.
Because I'm completely sure that everything is properly done, but it's nice to see this, because I think ethics in research is important. Okay. So this said, again, congratulations for the work. Well, I must have posed some questions. So the next question is, what part of this work did you personally? Have you tagged the fish?
No.
Or
So I
witnessed it?
no. So where basically I started is at this very extensive data set that I got. So it was 700,000 columns long, and I did a lot of exploratory analysis in the beginning to get like a head of what is the data, where do animals move. So I had took a very long time to really understand what it means, the different reef systems, the different interactions, and then to come up with with my supervisors together, some analysis, how to understand this. And of course, there was also some intricacies, because since we do not have, for some individuals, constant detections, or some just decided to leave the array, or some just died, we never know. There's a lot of analysis that we couldn't apply, that initially, like, we tried. So we just went with like exploring the data set and adjusting what we could analyze with it.
Okay, perfect. No, I totally understand that it's completely impossible to do all this work in 6 months. So assuming that it's completely impossible, I know that what you do normally is to inherit some work that has been previously
Yes,
done
exactly.
in the lab, in the host lab.
Yeah.
But well, I really advise you to go and tag or see how the fishes are tagged.
Oh yeah, I
Did
was
you
also
do
in
it?
the fields. I
Oh,
was also
great.
doing field, but it was not for my project. So
Yeah, but
the Australian Institute of Marine Science took me also in the fields, and I had also done my field work experience, and we also tagged whale sharks, and so
perfect. It's very good. You know everything since the
yes,
very
yes,
beginning.
yes, I took the part from back then.
Very good. It's excellent for your formation, of course, that you
Yes.
know all the steps in the
Yes.
protocols and procedures for your
Yeah.
work. Very good. So the last question is about statistics. Here you did not present any statistics. I see this is an observational study, so in principle there is no much needed, but in case you want to compare the results between
To say or to say that there is partition or there is not partition.
exactly. So
There's a lot
what
of statistics.
numbers or what
Yeah,
tests
there's a lot of
did you apply?
statistics behind. I just didn't want to butter you with that. So, but there's a lot of statistics behind.
Good.
So we divided the whole data set. So, okay, maybe one thing that I'm going to highlight here after we used the raw detection data, we spatially use the data with spatial metrics. But to do this first, we needed to calculate the centers of activity. So if you imagine, always the pings of like all the fish are always at the receivers, right? So obviously it would be a strong bias towards the receivers. So then we had a 60-minute time frame where we meaned all of the detections and created this location data of the actual fish every 6 minutes. So this is the first step. And then also we distributed the space use of fish and of sharks into month, into day and night, and into hours. And therefore we tested with ANOVAs and with post-hoc tests if there's significant difference. And we found a significant difference, for example, day and night. There was a significant difference for a larger area use at night. And also for the hours of the day, we distributed it into 4 categories to do the ANOVA. And we found in the afternoon and in the evening also larger use areas. And this also goes with the nocturnal feeding behavior, maybe to expand the niches, and also the raw detection data, which showed the activity of the raw detection or correlated with it. And yeah, and one little thing that maybe we can see also here, that they actually migrate very far in some months. So it's most likely spawning behavior. And yeah, so we could also see, like, of course, significant difference in the red bass behavior over the different months, where you can see already here, they just leave.
Yes, they disappear.
They disappear. And also the patterns could be lunar cycles, because it's always like a bit, so we want to investigate this also further.
Laura, sorry, it's not happening with shark.
It's not happening with sharks, no.
So there is no any nursery, or this is the nursery area.
I wouldn't go.
I'm chatting with her because we work together, so.
Yeah, so we share the question.
Yeah. No, that's fine. Like I wouldn't assume it like this, but so what we saw is that the sharks are just very localized at these areas, and that also means a lot, because these areas, as you saw it, they're very far off everything. So they basically don't leave a lot. So this is so far off that they're somewhere in nowhere. So looking at atolls, reef atolls, is very different than looking at something at the coastline, or where nurseries are usually. So because they basically stay there. That's also why this is such a good natural experiment, because they don't move far. So we don't detect any sharks from here to here or something. That's not happening. So these reef sharks are really like resident in these areas.
And the other individuals, the other species comes back to the same reef. So it
Yeah,
uses
no. So
or not?
meaning that the red bass travels a lot. I mean like 10 kilometers or something like this.
Yeah.
So that's what I mean, travel a lot. So they go basically from the rolling shoals. The red bass goes. So here the red bass has a very small home range. So there are fish. They're like little reef. So they're turning around here, mostly here, and then once a year they're going here, or twice a year they're going here, and then they go back. So this is for their size, for their movement. This is a large extent.
Okay.
So the sharks, they travel definitely more far. So the scales are different.
Okay, just we are just almost on time, but so I will make only one question, but I think that is very important. It's very important for you, Laura. The first thing is I like delighted with your presentation. We are very proud when we saw the Oviedo team presenting again because it's fantastic, to see all that development, professional development. But what is the role of partition studies in the conservation of those species? I mean, this is something that is important for people that are not ecologists. For example, in my case, I am geneticist, and when you are talking, I'm thinking about they took samples, for the sharks to see the levels of lateness of the shark. So I'm thinking in genetics, but it's important to stay. Why these studies are relevant for the conservation or the management of these species in this particular area. It's very important to put the context of the research, because I was very surprised and delighted with the distribution of the cohort, but what is the meaning of that? Why is this information useful? It's important to state that. So can you comment something briefly? Yeah.
Oh no, but I thought maybe I didn't make it this clear. But obviously, so when we have spatial partitioning, we found this spatial partitioning at an unfished reef. So this means always this talk about okay, could maybe be red bass and sharks, could they be functional redundant? Could they overtake the niche or something? This is the idea of when we fish all the big fish of a reef and all the sharks of a reef. So what happens actually to the reef? And now we see that in a healthy reef ecosystem, we see spatial partitioning. So we see all these mesopredators keeping their trophic roles. And this is actually what we need. So if we have a shift in this and we see that suddenly, they completely overlap, they prey on the same things. And then we make, okay, there's something off, because rather there's not enough sharks, there's not enough food, so something happens there. And then whole trophic food chains can get out of control, but we don't know how strong we actually mitigate this. So of course, thinking about how we can reduce the fishing pressure and how we can do this would be the first and best solution to that.
This is very important. Thank you so much. You are done. 15 minutes. Okay, we have Lizanne. You ready?
Yes. Maybe.
Let your own line.
Okay.
Okay, the floor is yours.
Hey, wonderful. Good morning, everyone. My name is Suzanne Kippers, and today I would like to give you some insight about microfragmentation as an active restoration method for Madrastic cactus, which is a prominent coral species in Brazil. This is also where I conducted my master thesis research at the Center of Marine Biology, the CIBIMAR of the University of Sao Paulo. As a lot of you might know, coral reefs are highly productive biological ecosystems that provide numerous ecosystem goods and services, including biodiversity, tourism, fisheries, shoreline protection. Unfortunately, they are known to decline globally due to anthropogenic stressors, notably climate change. In this map, we can see in light blue the 12 reef areas of the world that are predicted to shift from 84% of refuge reef areas globally to 99.7% of exposed reef areas, with an increase of 2 degrees Celsius. This is impacting the natural recovery of reefs, which is diminishing due to the increasing intensities and frequencies of the disturbance events. It's impacting the total life cycle of corals, from early recruitment to reproduction. And to address this issue, a lot of restoration methods have emerged. Prior passive habitat protection has mainly focused, prior marine conservation has mainly focused on passive habitat protection. However, recent studies have shown that we need to combine active and passive habitat protection. Active restoration methods are mainly used where passive habitat protection reach their limitations, like creating MPAs or no-take zones. As active restoration methods, we define anything that is aimed to aid ecosystem recovery or enhance its resilience. It can be either sexual or asexual propagation methods. One of the most common restoration methods is coral gardening. It's a two-phase method, which contains of the nursery phase, can be in situ or ex situ, and is followed by a transplantation phase or outplanting phase. This common technique has been tested for a lot of species and location and showed an overall survival rate of 68 to 95%, regardless of the location. As I mentioned, nurseries can be ex situ, so land-based in aquariums, or in situ, on the reef, in the field directly, or in the water column. The outplanting is the outplanting of fragments to designated reef areas. This can be the second phase of the coral gardening technique, or it can also be direct outplanting, where fragments are affixed to artificial substrates and then outplanted to the reef. Prior coral restoration has mainly focused on fast-growing branching corals, which, however, tend to be less resilient to disturbance events and especially bleaching. Microfragmentation is a technique to go against this, and it focuses on massive and encrusting corals. These are largely underrepresented in coral restoration studies, with only showing 5%. Microfragmentation is where we take mature colonies and cut them into small, mature colonies of massive or encrusting corals, and we cut them into small pieces or fragments between 1 and 15 square centimeters. This induces growth rates which are much higher than the normal lateral extension in the field. It's known to be associated with the wound healing response, more precisely with the disruption of calcium homeostasis. And if we place fragments from the same donor colony close to each other, the growth rates can be even higher until fragments fuse. This is for the benefit of shared resources and spatial competition. Microfragmentation uses the techniques of coral gardening, and it's mainly a nursery-based technique. But when we're looking at costs, it's very interesting that studies have found that it can also be direct outplanting with success. So costs in restoration projects mostly vary due to nursery type and time. And it's a trade-off that has to be very specifically looked at for each project for a location and species, because the performance in nature due to competition and predation can be unfavorable for the corals. When we look at overall coral restoration studies, they're largely concentrated in the Indo-Pacific and in the Caribbean. As we can see, there's only very, very little studies in the Southwestern Atlantic. When we compare the Brazilian reefs in the South Atlantic to the Indo-Pacific and Caribbean reefs, we see that the water is very turbid compared to oligotrophic waters, and that we have mainly massive forms of corals. In general, we have high nutrient loads and high sedimentation rates, with low coral diversity and cover, which is usually between 2 and 10%. Madrasas de cactus is one of the most widespread zooxanthine corals in Brazil, and it has a wide latitudinal and depth range, occurring from 0 to 100 meters on vertical shaded walls and well-lit, shallow, flat surface areas. It's a coral that varies very much in its ecotypes. It can be nodular corals, coral colonies, or massive and encrusting forms. It's a hermaphroditic coral and a brooding species, and although it has this wide abiotic range, it is very vulnerable to temperature extremes and high sedimentation rate. That's why it's unfortunately predicted to decline by 80% until 2050, and therefore listed as critically endangered. Madrassus cactus has previously not known to be a reef-building species, but however, close to my study area, which is here, the southernmost biogenic carbonate reef in the Atlantic Ocean has been found, and as you guessed it, it's mainly built by Madrassus cactus, which is displayed here in yellow. So, due to the global reef decline and the predicted issues that Madrassus cactus might face, we wanted to look at the understudied area of the southwestern Atlantic, and as a kickoff study, look at the ideal initial fragment size of outplanting. We wanted to compare the performance of the corals in ex situ and in situ nurseries, and test if direct outplanting could match the ex situ rearings. As I mentioned before, my study was conducted in Sao Paulo state, in the channel of São Sebastião, which separates the island from the mainland. For my study, we had one sampling site on the coast of the island, 3 in situ outplanting sites, 2 of them on the coast of the island and 1 on the coast of the mainland, and 2 ex situ sites at Sibimar. For my experimental design, we cut the corals in 3 different fragment sizes. The small consisting of 4 1 square centimeter pieces, the medium consisting of 2 2 square centimeter pieces, and large of 1 4 square centimeter pieces. On each face of the pyramid holder, we glued these 3 fragment size classes, creating a replicated block design, but by outplanting 8 holders per location. As I mentioned before, we had 5 locations, 3 in situ and 2 ex situ sites. We took the holder design inspired by Knapp et al., and we specifically chose this holder due to the inclination of its sides, which would reduce the sediment retention on the corals. For my workflow, we looked at the sampling site. We went to one sampling site and sampled with hammer and chisel 15 colonies. We brought them back to the laboratory where they faced the quarantine phase for 1 or 2 days. Then we microfragmented them in the 3 different size classes, and after that, we weighed them in the size classes and glued them on the holder. In total, I had 40 different holders, which were randomly distributed along the aquariums, and then had an acclimation phase of 1 week. After this, I took pictures for further analysis and outplanted in the in situ sites by looking for flat rock surface areas, scraping them and cleaning them of any fooling organism, attaching the holder with epoxy, and then outplanting the holder for 90 days. Same for the ex situ outplanting. We had 1 outdoor covered nursery and 1 indoor laboratory nursery. Both of them had 2 herbivorous crabs for cleaning practices, with feeding twice a week from a nearby bay with plankton. For the indoor laboratory, we also had a temperature control and oxygenated water with air stones. As I mentioned before, my experimental monitoring period was 3 months, so 90 days, and we took the response variables at T0, so the beginning, and T1 at the end of the experiment. Therefore, we weighed the fragments, I counted the polyp number in image J, and I counted the 2D area, calculated it in image J, and I took 360-degree angle pictures to create a 3D model with Agisoft Metashape. This I didn't include yet because the results are still calculating at the moment. To characterize the different in situ sites, we also looked at some environmental parameters. We outplanted sediment traps for 1 week at 3 different sampling times, and we took seawater temperature with eye buttons. Coming to some preliminary results, as you can see, we had an overall high survival rate of 87%, with the ex situ sites displayed in green showing higher survival rate, as we expected, due to no competition and predation, and in situ we lost some fragments. Due to the fragment loss, we interpolated the data where we had partial fragment loss for the small and medium-sized classes, and also due to fragment loss, we were not able to use the replicated block design per holder, and therefore we tested for nestedness within holder. We couldn't find any, and we used the average per holder approach. Then we continued with for the significant differences, we found significant differences for all 3 response variables for location and fragment size, and we found significant differences for polyp number in the interaction of location and fragment size. For further analysis, we looked at plant comparison. So we compared all in situ sites with all ex situ sites. We compared in situ sites high sedimentation rates with low sedimentation rates, and we compared the two ex situ nurseries. Looking at the plant comparison mainly now, we found only differences for weight compared to in situ and ex situ, with in situ actually showing higher growth rates. And the low sedimentation site, Sao Pedro, was the best in situ site. And we didn't find any significant differences in most of the parameters for the ex situ nursery site, only for weight. Again, here, surprising, the low maintenance showed better growth rates than the high maintenance. Now I want to take you to a little example. We use post-hoc testing also to compare the different fragment sizes, and I want to go to the example of area. Here's some preliminary data visualization, and we can see a clear trend from large fragment sizes to small fragment sizes, increasing growth rates in all stations. Here's shown the area growth rate, the percentage change per month, and this is also coherent with the results that we found in the post-hoc testing. In general, this is also coherent with other studies, which showed that small fragment sizes had higher growth rates. For the plant comparison, as I mentioned before, we only found significant differences for Sao Pedro, the low sedimentation rate site. Coming to my conclusions. This was the kickoff study for the Southwestern Atlantic, we were very happy to say that we were able to successfully microfragment and outplant Madrastis the cactus for the Southwestern Atlantic. We had a high survival rate regardless of the fragment size, and in all station and fragment sizes, we had overall positive growth rates. Coming to my objectives and aims, with the preliminary results I have now, we can say that the small, so 1 square centimeter, or medium 2 square centimeter pieces are the ideal initial fragment size. We compared ex situ and in situ sites, and we didn't find any significant differences, which means that direct outplanting is definitely the way to go for Madrastis the cactus in the Southwestern Atlantic. In general, this is the beginning of active core restoration in the Southwestern Atlantic, and it's just a snapshot in time, so only a 3-month study. So this is very important to say. For further studies, longer study periods should be conducted, and they should also include ecological monitoring and ecosystem-based management, like creating passive habitat protection, like MPX, to mitigate anthropogenic pressures. And with this, I would like to thank you for your attention. Do you have any questions?
Thank you so much, Susan. Okay, so Eva, you start now. Yeah. Okay, yeah. Any questions for the others? Short question?
I was just wondering, when you have the small fragments, do you expect the growth rate to slow down when they eventually get to the size of the larger fragments?
Yeah, so in the studies that I compared to, the high growth rate was mainly in the first few months. This is a very important thing to say. But in general, it's like looking at the large scale, if we have a lot of small fragments that we get from a mature colony, we have more potential for growth rate. And then if they reach the large fragment size, we already have more tissue compared to only cutting large fragments.
I was wondering how you determined your fragment sizes that you chose.
I looked at different literatures, and also this, as I said, the colony shows very different ecotypes. So it was actually quite hard to get to the 4 square centimeter size. So larger was definitely not an option.
Okay.
Yes, thank you, yes. So again, thank you very much. Excellent presentation, really. And also for a very, very good work with a lot of implications for conservation.
Thank you.
Even being Jiminy the Cricket, this is excellent. I have no questions about the ethics committee, etc., because it's clear you are working for restoration of a critically endangered species, which is simply great. So my first question is, I assume from what you told us, but you haven't submitted yet. You didn't submit
Yes,
yet.
I didn't submit yet. I'm still in the process of doing my statistical analysis, further analysis. This is just some preliminary results. Unfortunately, there was a heat wave and also a storm, and the filters of my nurseries blocked. So half of my experiment died, and I had to do it again.
Oh no,
Yes,
it was longer than expected.
way longer. Because I was already like 1 month in, and then it died, and I had to start everything again from the collecting to the excavation phase and taking the pictures again.
So you did everything
Yes.
yourself. Oh,
Yeah.
this is a lot of work.
Yeah.
But even though you have already some results and statistics seems to be quite
Yeah, this
sound.
is also a big thank you to my supervisor, which helped me and pushed me a lot. And yeah, definitely big thanks to him. And in general to the working group. I got a lot of support from people helping me to outplan the holders and also to weigh the fragments. And yeah.
It's so good to.
Yeah, really good community. I'm very, very happy and very thankful for this.
Very good. I think from your results, even if they are preliminary, it seems like the strategy, this strategy, it's good and it's valid for restoration, the microfragmentation.
Yes, and also like for the Southwestern Atlantic, because it has never been tested or studied, so we were very not sure if it would work, especially like regarding survival rates of the fragments, because it's very turbid waters, and my study site was in a channel, so it's extra sedimentation and also runoffs in the channel, so.
Yeah, and both in situ and ex situ can be efficient.
Yes, and I mean the amazing results here, the
Yes.
most protected site showed the highest overall growth rates for all parameters in situ, which means we can skip the nursery phase and directly outplant the fragments, which is amazing in terms of upscaling the restoration projects in costs. Yeah, and also like in like work time in general, like which is of course related to cost.
So since we have to pose you some question for you to invent or something, just for you to create, to develop on this, I'd like you to comment a little bit or to develop a little bit about the real need for this restoration measurements. From your perspective, what's better? To work more for prevention, I mean, or conserving the environment, the natural environment, and let the species to manage themselves, or to do this type, or to put more effort instead of marine protected areas or protecting the environment, to do more effort on this. What is better for the conciliation?
This is a very good question, and I must say I'm for both. Like I think no, but because when we only use, as I said in the introduction, when we only use passive habitat protection in areas where this definitely has reached their limitations, we have to help and aid the ecosystem with active restoration methods a little bit. To create maybe some spillover events also to other areas, and yeah, like just support with this. And I think it really depends which area, like which location, and what's the exact focus of the restoration project. But yeah, I think we're so far in climate change and other anthropogenic pressures that one is mostly not enough.
Okay, so probably conciliation is the word. Okay, thank you very much.
Thank you.
Thank you, Eva. Lawrence?
Well, I congratulate you and I thank you very much for your nice presentation this morning. We like it. Yeah, it's the best room, that's for sure. I think your study is really interesting, and I have some question regarding the difference between what you observe when in situ and ex situ, because in fact you said that there is the water is very turbid and there's a high sedimentation rates in the water column. So I am wondering if you have an idea of the nutritional status of the coral that when they are inside in comparison to when they are ex situ. If you have an idea, or if you had to address this question, like, is the feed of the coral good enough in comparison to what they get in the channel?
You mean what we fed them?
Yes.
I think so. I mean, we did it to the best of our knowledge, and we used plankton nets to collect plankton twice a week from the bay, and it was usually a quite high concentration of plankton, and we stopped the water flow for 1 hour in that time where the corals fed, and then after I turned back on the water flow. So
and yes, and if you had to question, like, I imagine I will play the devil, and I say, oh, that's not enough. They are not fed enough,
Okay.
or this is not their favorite prey, or they are lacking. I don't know. And how would you test that?
Ideally, we would test it in different with different food and different nutritions and different concentrations in ex situ nurseries, and compare the results. But yeah, unfortunately, like I found in the literature that usually a lot of ex situ nurseries use Daphnia to feed the corals, but yeah, time was limited for me, and I already had a lot of work to do. So collecting the plankton from the nearby bay, also to close to the site where we actually sampled the corals. I'm pretty sure that this is what they feed on, and yeah, for the amount of food that we provided, we would have to do further testing and further analysis to see if it's really enough for them and if it influences their growth rates.
Because there is definitely a relationship between food and growth.
Yes, definitely.
Or feeding.
Yeah.
Feeding.
And but I must say, like, they did some studies in another working group before where they tested for bleaching in different temperature conditions, and they used the same feeding techniques. So I used their knowledge and their expertise to do my experiments.
And also regarding the strategies to like, imagine if you have a large program to do this restoration, and you would have to pick up the location where you put the
holders.
Yes, the holders. How would you locate them in terms of circulation? Because you have this channel that probably is acting like a funnel, and then let's say a flow of particles, not only of water for the corals. So you have if you had to select location in relation to water circulation and also water temperature, would you locate the transplant in the same location than where you get them off, or would you try to put them like maybe a bit deeper where the temperature will be predicted to be lower?
I would like to show you something, but this is not working anymore. Does anyone know how to do this?
It's
It looks like it wants to record something.
yeah,
Sorry.
the password. it needs, no, no.
No, but
Nothing wrong with you.
about that, and,
It's a,
no one has the problem.
no, I don't know what
You don't have.
what is the password? I will try. I will try one.
Yeah.
In the meantime,
Yes.
are they taking samples of genetics for genetics studies? I
This
mean,
is-
to see
yeah.
the level of kinship or relateness among what you are planting or no? Or what?
I would have really loved to include genotypes in my study,
Yeah.
but as this study was already quite extensive and including genotype, meaning we had to triple or
This is
Why-
another master project.
yeah. But it's definitely a thing that influences growth rates a lot, and we are aware of this. And yeah, sadly, there was only 6 months to do it. So, what was your precise question?
No.
Oh, okay,
I was just
okay.
asking about if they, the group, was collecting samples also for genetics or
There's
studying.
another working group
yeah.
that works mainly with genetics
Yeah.
and looking mostly at Mussus media hispida, which is the other coral species which occurs in the area where I work in, but also now on Madame Fisticulus.
Yeah. Okay, I have, yeah.
I'm
We are
sorry.
on time. I don't know if
Come and unlock the screen.
Okay. Yes.
I just a philosophical question before finishing. Yes, marine protected areas and restoring the habitat. But
Yes.
in our experience, I mean, in Asturias, for example, it was a lot of years supplementing the salmon populations and putting everything in the river. But then at the end, we discovered that restoring the river was the best.
Yes.
And
sorry,
this-
I'm sorry.
Yeah, restoring the habitat is the best. I mean,
Yeah.
the rest is just to waste money in the river. But yeah, I at the end, when I think about okay, restoring the corals, but climate change is something that is difficult to stop. So my
Yeah.
question is there some hope from the research community of corals that that this restored will be more resilient
Oh,
and not
Maybe.
Don't try with.
Okay, sorry. I will.
no, that if there is some hope about if habitat that have been restored will be more resilient to global climatic change than others, are there any evidences about that?
Yes, I think like looking especially at genetics and specifically targeting species that are genotypes that are more resilient and using them for restoration purposes would be a first good thing to do. And in general, like the Southwestern Atlantic is a very different reef area compared to others. So it has this turbid waters, and the corals are more robust compared to tropical corals. So I think with the turbidity, for example, the UV doesn't make such a big effect on them. And also, thank you. So I think there's hope. I think we shouldn't give up. Like if we give up, there's nothing to do for us anymore. If we just say, okay, everything's going to go down anyways. We have to hope and try our best for now, at least. I mean, we can't change what's coming for the future, but to act now and local in small scales is a big step already, at least for these ecosystems. And then we have to see what the future holds for us.
What do you want to show?
I wanted to answer your question. Can you repeat? Sorry.
about, you know, I asked you about the if you had to transplant or
Yes.
to pick up a ideal site,
Yeah.
and about the temperature in if you would risk in fact to extend the area in places where the temperature is lower in order to counteract the global change.
So as we can see, this is the sampling area, and this is the site which had the best overall growth rate. So first of all, it's very close, and it's mainly outside of the channel. So the other station at the coast is very close to the channel, and the one at the mainland is also like, if there's a southern swell coming, it hits directly on this part. So it's very exposed sides. And yeah, I think if you outplant corals, you definitely have to check for the location first and see what are the environmental characteristics of locations where these coral could grow best. In my case, we found like protected bays with low sedimentation rates and lower temperature are the best to. Sorry. Yeah, here are the preliminary results again for the sedimentation, like the log of the max flux. And as we can see, Sao Pedro had overall the low sedimentation rates. And also if we compare the second sampling time, which had where there was a storm, this site was really, really low compared to the others. So it's way less exposed to like re-exposure of sediments. And yeah, I think this is one of the most important things to consider when you do a transplantation study.
Okay.
Okay, Lisana, we are very relaxed because we have only 2 presentations, so we have time and we are talking, but you are done. You are mastering time. We see you at 11.
Impressive.
Very, very good.
Super interesting.
Seguimos aquí, ¿no? Yo soy Real, como esto ahora no está bien.
Sabes que ahora no está bien, entonces no sé. Yo creo que voy a 4. Ahora voy con Gómez. La primera vez que voy a
Yo estoy aquí.
Okay, let's start. Okay, so Fellini is talking about enhancing coastal biodiversity for nature-inclusive harbor infrastructure. Okay, the floor is yours.
Hey, good morning, everyone. My name is Apsara, and I'm here to present my research on enhancing coastal biodiversity through nature-inclusive harbor infrastructure, focusing on a case study at Port of Vigo, northwestern Spain. So my supervisors are John and Maria from DPUFA, and the study investigates the potential of novel concrete structure to mitigate the impact on biodiversity to the coastal development. So the coastal development has been searched globally due to its strategic importance, leading to expansion of harbors. Even though this expansion gives numerous social and economic benefits, it also leading to environmental challenges. The construction and expansion of harbors disrupt ecosystem and degrade habitats and threaten biodiversity at the end. And also, because of the climate change, the rising sea level also threatens this coastal habitat, leading to coastal squeeze. And in these two illustrations, you can see the coastal habitat during high tide and low tide and how they are important for the life cycle of marine organisms. And the coastal habitat like mangroves, seagrass beds are very important for marine species. They are providing numerous ecosystem services, especially they are providing breeding and foraging ground for several commercially and recreationally important marine species. So the degradation of these habitats are really bad for the environment and also for the economy. And when we consider about the Port of Vigo, there are major two types of harbor infrastructures, seawalls and strip traps. Seawalls you can see in the first picture, the seawalls. So seawalls are vertical or near vertical structures built parallel to the shoreline for the coastal protection. And strip traps are large stone boulders which are placed along the shoreline to prevent soil erosion. In these two pictures, you can see how this traditional harbor infrastructure disrupt the life cycle of marine organisms. And in this study, we are trying to replace those traditional harbor infrastructure by nature-inclusive harbor infrastructure made out of eco-concrete, and it will have a less impact of the habitat. So the study has three main objectives. The first one is that novel concrete infrastructures or the eco-concrete structures has higher taxonomic diversity than the traditional ones. And the second one is the eco-concrete structures has higher species abundance than the traditional harbor infrastructure. And the third one is the novel concrete structure will provide new habitat during the low tide, which is absent in traditional harbor infrastructures. So when we move to the methodology, the study was conducted at Port of Vigo, northwestern Spain, and the study employed before-after control effect or the Baki design to see the effect of the novel concrete structures before and after installation. And for biodiversity monitoring, we used remote underwater video systems. And this is the timeline of the study. So there were 3 monitoring periods. As the baseline monitoring in fall month of 2022, we did the pre-installation sampling before the installment of the eco-concrete structures. And during January to March of 2023, those eco-concrete structures were placed in the port. And during spring month of 2022, the first post-installation sampling was conducted to see the immediate impact of these structures. And during the fall month of 2023, it's exactly just after 1 year of the baseline monitoring, we did the second post-installation sampling to see the long-term impact. And when we consider about the study site and the structures, there were major 2 study sites, Port Cultura and Bausas. And the analysis was conducted separate for these 2 sites because these 2 sites are completely different with the environment and everything. So in the Port Cultura site, we had 2 eco-concrete structures and the traditional seawall as the control. When we talk about the Azure seawall. It featured the square pattern with different height levels. So it was intended to create microhabitat for the marine organisms. And the next one is the mangrove seawall. It was trying to resemble the mangrove roots, which provide numerous habitat for marine organisms. So for each eco-concrete structure, we installed the traditional seawall nearby at the same time to compare the difference. And when we consider about the BOSA's study site, the normal coastal protection was traditionally achieved using the riprap's or the stone boulders. So we replaced those riprap's using like a concrete structure called coastal dock. But it's unlikely the horticultural site, the control or the riprap's were already colonized. So it is kind of a limitation of this. And the coastal dock, it has a pool which try to mimic the natural rock pools, and it can contain like around 100 liters of water. And for the biodiversity monitoring, we install underwater video cameras in each study site. It was equipped with a timer, and it was programmed to record 2 minutes every half an hour in daylight hours. So apart from the main study, we also installed cameras inside the coastal tidal pools to see the difference during high tide and low tide. And from all this, so we collected 3,973 2-minute recordings from the main study and 102 minutes recording from the coastal low tidal pool study. And we used this data to measure the species richness, diversity, and abundance. And the video analysis was conducted manually using VLC player. So the key measurement was the max N, or the maximum number per species observed during these 2-minute recordings. And we try to identify all the species to lowest possible taxonomic level. And the statistical analysis was conducted using RStudio or the using vegan package. We measure the species richness, Shannon median index, and system diversity. And we use generalized linear mixed models to compare the effectiveness of these eco-concrete structures using GLM-TMB package. And when we look at the results, for the richness study, we try to select few focal groups. So there are 6 focal groups. And when we look at the season, so we can see a significant negative difference with the baseline monitoring data on the spring 2023 data. And when we look at the treatments, mangrove seawalls shows a significant positive impact on both species richness and diversity, but the Azure seawall did not show any significant compared to the traditional structures. And when we moved to the Busa site, it was the same with the spring 2023 data. There was a negative significance with the baseline monitoring. And we look at the treatment, the coastal lock shows a negative effect, mainly due to unlikely in horticultura, the control or the itself were already colonized. So I think for the Busa slide, we need longer monitoring time to see the actual impact of the coastal loss. And when we look at the results from the coastal low tidal pool study, so we saw during high tide, the species abundance and richness is higher than the low tide. But even though during low tide, we saw there is a clear evidence for the species persistence, like shrimps and gobies were there. So it indicates the creation of new habitats, which wasn't present in the riprap's or the And when we move to the discussion, so this study shows the potential of nature inclusive design to mimic the habitat destruction by the coastal development, especially results from the horticultural side, especially the results of the mangrove seawall shows that they support this idea. As the second point, when we consider the results from the horticultural side, there were 2 seawalls, mangrove and Azure, but only the mangrove seawall shows the positive impact. So it highlights the importance of considering the specific defining of this for biodiversity enhancement. And as the next point, even though the coastal of the structure shows initial negative impact, it shows it has potential to create new habitats. So it is similar to natural rock pools. In some studies, they found like the gobies are also present in natural rock pools, which is kind of similar. And as the last discussion point, both horticulture and Musa style need longer monitoring time because the structures were only like there for like 7 months. So I think it's need more longer monitoring time. And it also, some species may be relocated to new areas because of the initial habitat destruction, and it will need time to recolonize those new structures. And as the conclusion, so this study shows the modified infrastructure has an effect on mobile fauna. In the literature, there are like supporting evidence for the sessile fauna, but this study shows it has also impact on the mobile fauna. And as the second conclusion, the results from the horticultural side, especially the mangrove seawall, shows the importance of considering the specific design for the habitat enhancement. And as the third one, this study provides shows the potential of using new structure to create new habitats during both high tide and low tide. And this is my acknowledgement. I would like to thank research team from DGWAC and DG Sustain, and you can see this is a collaborative project and this is kind of a part of it. So I'd like to thank everyone here. And that's all.
Okay, thank you for the very nice and clear presentation. So, okay, question. Who wants to start
Okay,
first?
we start. Thank you very much for your nice presentation, beautiful slides and drawings of the fish. I would like to have your thoughts on the. I mean, if I understood well, you have compared two different sites. I mean, one control and one site, and I would like to have your thoughts on the choice of your control and the number of replicates, because you have followed one site with the e-concrete system compared to a control, and I would like to have your views on the fact that you have one control and the choice of this control and the number of replicates.
Okay, actually, we have 3 controls. For the accuracy role, we had 2 controls.
Okay.
So I combined everything for the control. That's why, like, yeah.
Oh, I missed this.
Yeah, but I didn't
information.
Okay. Yeah, I didn't say.
You mentioned beyond Bashib design. Okay,
Yeah.
Okay, that applies at least to
Yeah, and I have this is the sampling port, and so we have this much of cameras in like every each and every site.
Okay, yes, sorry, I missed that in your presentation.
It was not in the presentation. I put it at the end because it is.
Okay. So they were all distributed along the coastline. What industry control? What can you tell us then more?
In the horticultural it's more deeper. So it's like, it's a deeper area, but the Bosa style, it was a coastal area. So that's why we separate 2 analysis, because it is different.
Okay, and if I well understood also, you mentioned that there has already been studies on the sessile organism. So what can you tell us on what link you would make between this study on, I would say, I guess it was invertebrates, or what type of sessile organisms?
Mainly the shell organisms.
Okay.
It's attached to the seawalls.
Okay. So what link would you make to try to link those past studies with your study, and what was your hypothesis? Initial hypothesis based on those previous findings.
I didn't connect these 2, but with my results, I can say like both of them have connection, because in my study it shows like the mobile fauna also has a positive impact on the biodiversity, like the previous studies about the fauna. Yeah, but it's not in my hypothesis.
Okay, but I was thinking, I mean, if fish would come, why would they come?
Because from the observation, we saw a lot of vegetation in these new areas. I think that's the main reason. It's not like in the control areas, there was, because we placed in the horticultural area the control site at the same time. So compared to the control site and the treatment site, in the treatment site, there were a lot of vegetation. And I think the food availability and all these things were the reason.
So food and protection. Okay, so because there was more also invertebrates, also on those.
Yes, but we didn't measure those things.
Okay, thank you.
Thank you. Thank you. Very interesting presentation. I like the fact that you use the Baki design. That's very good. I have a question about the timing of the choice of the timing you of the sampling, September, October.
Yes.
Because we've done some work in harbors and stuff, and really marinas and harbors are hotspots of biodiversity, because especially for juvenile fish and as nurseries, because they offer a lot of structure protection, and there's a lot of algae growing and stuff like that. But in September, October, that's not when the peak period of or at all. I mean, the recruitment I would expect in Vigo would be in the spring and summer. Yes. So what you are counting is mainly larger individuals. So you're missing out on the potential as a nursery habitat, if you like, or
it was a limitation because the project was already planned with all the collaborators. So we only had funding for this month, so that's what happened. Yeah, I would like to do a sampling again in the spring to see.
It would be very interesting to see as a source, and to compare, the more the three-dimensional aspects of the habitat. Have you heard of these biohuts? No, didn't come up.
I think I heard.
Yeah. This is a French company that is producing these artificial habitats to put inside marinas and ports, where you have a structure, a wire structure cage, and inside you have oyster shells inside another structure. So the outer structure keeps away the predators, and the small fish can go inside and hide in the coral, in the oyster habitat, and it's extremely effective as a nursery habitat for juvenile fish. That could be something to employ, maybe in test in there. Yeah. So you didn't find so many differences between the original wall and one of them? No, which one was the one that had
the positive that had from the mangrove seawall, the one in the middle?
That was the best in terms of the. Okay. What's so different from the Azuri seawall? I mean, in terms of structure.
It's in the Azuri seawall, it's just squares with the different height levels. Mm-hmm. So, but this one is more try to mimic the mangrove roads, but it is, I think it's just the shape. It's just the same like the concrete mixture, but it's just the shape.
Okay. Yeah. Do you think also that it's a question of time? You need time for the community to mature, to the algae to grow, because the diversity will certainly that will attract more fish.
I think I recommend to do a research after 5 years or something to see the actual thing happening there.
Also, I noticed. Can you go back to where we have the main species of fish?
The fish.
Yeah, that one. Yeah. So. So, for example, sand smelt, Aterina, is really a pelagic fish. They wouldn't be really attracted by hard structure. They're just swimming along there. It's not. I don't think there's really that effect. I wouldn't expect much difference. Gobies, yes, because they would be on and they're resident. The same thing with gray mullet. They would be. Although you see, there's a difference there. It seems. So maybe there's more food for them on the. Yeah, I think it's too early to tell. I think you need more time for this too. to develop. But yes, it's a nice, a lot of work. Obviously, you did a lot of analysis of pictures.
Good baseline.
Yeah, yeah, very good.
Thank you.
Okay, thanks. Nice job. Okay, so let me be, okay, this place, Busas, is where exactly?
It's inside the port of Vigo. It's like two
Vigo,
sides.
okay,
Yeah,
so
yeah,
the Galicia.
yeah.
Okay, no, can you go back to the two pictures where you showed the pools? Okay, okay, nice. Yes, look, I agree very much with Karim. I would expect something like that to change in few years' time. I would expect some large brown seaweeds beginning to grow. Okay, so about this, okay, do you only consider the fish, thin fish, or even other taxa?
For this one, we consider about the shrimps because it was just shrimps and gobies inside
Okay.
low tide, so we had to consider the shrimps.
So, okay, you didn't consider seaweed at all?
No.
Okay, okay, well, that's a rare seaweeds are okay. Yeah, okay. Now, look, I think, I mean, this is an important point because, I mean, okay, we are the Atlantic Sea, the Galicia, so at some stage, some large, the brown algae will appear in there. And well, I would expect that at some stage they will make a canopy, so they will really change quite a lot the structure and the functioning of the ecosystem. But okay, look, this is a very interesting work. Okay, I see that this project was part of the European project. I was wondering if, okay, this type of infrastructures were also placed were also deployed in other regions.
I found one study in American University.
Okay. I was wondering if, I don't know, are you aware of what happened there? If there were.
It was the similar, like they are looking at the sessile organisms, so yeah, it has like a positive impact on sessile organisms. Okay. And it also strengthens the harbor infrastructure because like the plants are like attaching, so yeah.
Something
yeah, sure.
else. So if you were to do this in the period when there is recruitment, we're talking about very small fish, would this methodology work using the cameras to be able to identify the
small ones?
The very small ones?
We could never, we just like put them as non-identified fries.
Okay.
Yeah. It also we have a project to like develop program to detect like without manually to do any like we are doing another project, it's not part of mine, to like AI thing to identify the fish but not the small one.
Okay, great.
How long those three I mean, you followed 3 sites, right, with the concrete? So
2 sites.
How long had they been immersed? How new were they?
It's like around 7 months.
7 months. Yeah. Right. Okay. When did
it was
very early, but it's good baseline data, so you come back in a few years and show that it's efficient.
Okay, any questions from the audience? Okay, about this, look, a more general philosophical question. Look, what is your personal feeling about this matter? Do you think it is okay? I mean, the operations of this type are done in harbors or other urbanized areas. Do you think it is okay? What is your feeling? It is really worth it to, let's say, kind of renaturalize this type of habitats, or how do you feel it would be better to try to conserve natural habitats?
Yeah, but because I have example in my country. I'm from Sri Lanka. Is that
Okay.
yeah.
Yeah, sorry, but okay, so
Yeah, we are made in a port city, so we are constructing like a new area. And I think for like that kind of things, we should use this type of things, but we need the other controlling measures. Yeah.
I wonder if there are any considerations from the audience. This, so now, because I'll tell you, in the past, I saw the other the people that submitted proposals for projects to be renaturalize this type of because let's say to build the habitats for the habitat forming organisms, habitat forming seaweeds. That, in my opinion, didn't make any sense. But okay, we will have still 5 minutes. Okay, Sarah.
Do you think based on what you saw, do you think there's a different structure or a different design of structure that would be better?
For the baseline?
Yeah, and just for any of
Design.
Yeah, just the designs of like improving the structure for the marine organisms. Do you think, like with the different designs that are in this study, do you have opinions about optimizing them more or something different?
I think we should focus on this because
Okay.
the results, it shows like there is an impact of the
Yeah.
But it's interesting to see that in your case, at least on the short term of your study, actually the variation in the shape of the substratum seems to be more important than the height of the okay, which I mean it's not. Was not something obvious in general. Okay, the general concept is that the more heterogeneous is the substratum, normally one would expect a more biodiversity, but once again, I think okay, it is necessary to monitor and to see what will happen in time.
What can you tell us about, I mean, all the video you have viewed, how you were just in front of me?
In front of I had another person, so we both did it. She's the one that who did a lot of videos. I just joined at the end. It was a lot of time consuming, so I recommend like to create a program or something.
Time consuming, and how could you, I mean, for the species identification, size?
I had to train like 2 weeks for that, so So
you have high confidence on the identification and
because I had the fish earlier, so it was easier for me to identify. But I think the one who trained me, she told me like it took at least 1 month to train. Yeah, for me it only took weeks.
Okay, so like taxonomy.
Well, I guess the experienced fisherman will be the best person.
Thank you.
Okay. Any more questions or comments? Okay, please.
Are there any type of structures that work better for different organisms? Like I'm thinking here of like the type of organisms that would be in the harbor, so like the snails, the crabs, the different algae. Like I don't know if you know about the settlement.
Like I didn't do about the society thing, so I don't have idea. Yeah, it's just the mobile phone, like the fish. Yeah.
I wonder, okay, even though you looked at the fishes, maybe in your videos, did you see any crabs around?
No, crabs,
Okay, you just didn't record them.
We have recordings, but in my study, I didn't include them. Even birds, we can see that.
Okay, well, if there are no more questions or comments, okay, thank you very much. And then we move to the next presentation. Then we have Arjun. Okay, so influence of mature epibentic fouling community from colonization patterns in offshore wind farms. Okay, this is very interesting, even because in some way, it might integrate what we heard in the previous presentation. Okay, the floor is yours.
Good morning, everyone. My name is Arjun, and I will be presenting my thesis, that is the influence of mature epibenthic fouling communities on colonization patterns in new offshore wind farms. So as you all know, that there's a global shift towards renewable sources of energy, such as solar, wind, etc. The European Union, in its National Energy and Climate Plan 2021-2030, set a goal or a target for its member states to reduce carbon emissions by 40% in 2030. For this, they set a target of 32.5% for renewable energy. In response to this, Belgium set its target of 17.5%. So as I said, one of the sources of renewable energy is wind. Offshore wind farms are one of them, and they are better than onshore wind farms, for example, because they can be built larger, they are more efficient due to stronger winds offshore. And they are bigger. Belgium is a small country, as everyone knows, on the coast of the North Sea, and this small part is its exclusive economic zone. Currently, it has 399 operational wind farms in this area, with new offshore wind farm areas that have been designated. Belgium has had a traditionally sandy seafloor in which artificial hard substrate was added. As we all know, when artificial hard substrate is added to the seas, organisms start settling on it, and this can have an artificial reef effect. For example, as you can see, this is a rock that was added to the sea, and there's a lot of calcareous tubes made from the polychaete, from polychaetes. There is also an increase in the faunal diversity of the region. These are some of the macrobenthos that I saw. The amphipods, barnacles, polychaetes, etc. Studies have also shown that there is an alteration in the benthic community. So even though it can be seen as a positive that there is an increase in the faunal diversity, studies have shown that there's an alteration in the natural benthic community of the region, and that these also act as stepping stones for non-indigenous species. These here are a limpet called Crepidula, and this is an invasive oyster species, Cracospia. So what is biofouling? Biofouling is the accumulation of organisms on any hard substrate that is added to the water. So this could be, you can see them on ships, you can see them on the harbors, you can see them on buoys. And in this case, you can see them on the offshore wind farms. They form complex communities, as you can see here. There's a large variety of stuff. There's mussels, there's bryozoans, there's some tubularia, etc. They follow a predictable stepwise order in most places. But in Belgium or in the Belgian part of the North Sea, recent studies have shown that these communities have not stabilized. So usually it takes somewhere between 10, 11 years for a community to stabilize, but in Belgium, these communities have not stabilized even after 15 years. So I had two questions about these communities. Firstly, do mature communities accelerate the maturation of new communities? So what that means is when the first wind farms were added, there was a soft sediment area. So there was no availability of hard substrate species. So it must have taken slightly longer for these hard substrate species to reach the wind farms. But now when they build the new wind farms, will it be faster, the maturation of the community? And if so, what are the differences in the species composition and diversity? So for the materials and methods, first I would like to show you where we did the sampling. So these are the old wind farms where the construction started in 2008, 2009. This one, Belwind, is the one that I took for my old wind farm data that was constructed in 2009 and sampling was conducted in 2012. That is 3 years after it became operational. These two, Northwester 2 and Mermaid, were then constructed in 2020 or became operational in 2020, and the sampling was done last year in 2023. Belwind is about, all of these are somewhere between 40 and 45, 50 kilometers from shore. And the depth is somewhere between 30, 40 kilometers, meters. The Belgian part of the North Sea is not very deep, with the deepest going about 56 meters. So this is the general structure of the wind farm. I did my sampling in two places. One was somewhere in the middle at the depth of 15. Divers did the sampling at 15 meters depth, and at the base where there are natural rocks, so not concrete, that have been placed there as cover protection. So 25 centimeter by 25 centimeter quadrats were taken, scrapes were taken using a putty knife by the scientific diving team of the Institute of Natural Sciences. At the lab, they were then fixed in 6% formalin, and then I had to sieve them over 1 millimeter and then separate them into different categories, and then I identified all of them to species level. At the end, I went through 8 samples, 9 samples, and had over 120,000 individuals from over 55 taxa. For the statistical analysis, I did all the various indices followed by an NMDS, ANOSIM, and SIMPER. So first, for the diversity indices, I wanted to see if there is any change in the community or the diversity. So my data was divided into two different categories. Firstly, autumn and spring, because sampling was done both in autumn and spring, luckily, for the years that I had available. And rock here is the cover protection rock from the base, and scrape is the scrape taken at a depth of 15 meters. As you can see, the red ones that are the new ones in most places have a higher abundance, as well as the different indices show that there is more diversity, or there's a change in diversity in the new versus the old wind farms, signaling that there might be an effect of, that there has been some change, and this could tell us that there is some effect of the old wind farms on the new ones. Similarly, when I did the NMDS, it was to first see if there is any visual difference. And the closer the points are to each other, the more similar the communities are. As you can see here, for the spring scrape samples, these two are the ones from 2012 versus this one, which is from 2023. This showed me that there is some difference, and I had a low enough stress level to believe that this the NMDS appropriately described the differences. I then did an NOSIM that gave me a R value of 0.73. It was statistically significant, which showed me that there is some level of difference. For the autumn samples, I also saw differences. These two are the ones from 2012, while this is from 2023. This was not statistically significant, and I therefore didn't proceed with a simple analysis, which shows which species. Okay, yeah. This has cut off. Okay. Anyways. Okay, that's right. So I did an simple analysis for the statistically significant scrape samples to show which species actually made a difference in the communities from the 2 years. As you can see, these 5 species contributed to over 70% of the differences. This species is also known as dead man's finger is a soft coral. It was only found in the 2023 samples, while the others were found more in the this was only found in the 2012 samples, where the others were found only in the 2023 sample. Similarly here, these 5 species were contributed to 70% of the difference. As you can see here, Crepidula formicata is an invasive limpet which was found in way higher numbers in the spring versus the spring samples in 2023 versus 2012. Indicating, so this shows us that, as I said before, these can act as a stepping stone for different invasive species, and this shows that there has been an increase in the number of invasive species, or in the abundance of invasive species from 2012 to now. The rocks, on the other hand, while they showed a difference and had a high stress, had a very low stress level, the ANOSIM showed no significant differences for both the rocks, the samples. But it showed the analysis showed that there was some difference, and I think that we can attribute it, and it does point towards the old mature communities having some effect on the new ones. Unfortunately, there were some limitations for the data, for the study. My study was limited by the data that had been collected in 2012. As you might have seen, there were many instances where I didn't have 3 replicates, or I didn't have data and I had to work around it. The sampling for the new wind farms was only done once. It was the first year, 2023 was the first year where sampling was conducted. As I said again, the lack of replicates due to, so one of the newer samples didn't have replicates because the divers couldn't collect them due to bad weather conditions, bad visibility, etc. In my study, I forgot to mention it in the methods, but I had to remove the colonial species such as the different unicates, etc., because it was difficult to combine the qualitative and quantitative data, and while I originally wanted to do it in my thesis, I didn't end up having the time. For future studies, I would recommend doing a multi-year study. So here it was only a snapshot where I had 1 year from 2012 and 1 year 2023. So it does not give any idea about the community succession and how that will change. So if more sampling can be done regularly for the newer wind farms, it would give us an idea if there is actually an acceleration in the study. Also, a method that would allow colonial and the countable species to be combined Hydrodynamic models and larval dispersion might also help us get an idea how these species travel from one wind farm to another. So, oops. Yeah, so in conclusion, this was an important first step to see how the organisms spread from one place to another, even though more studies are needed in the future. There is a trend towards different species. So even though the data was not significant, you could see from the NMDS and the indices that there was a change in the communities. It shows us that that we have a very limited understanding of marine communities, especially the macrobenthic ones, and that there needs to be a continued monitoring of these communities, because even though, for example, Belgium has been doing monitoring of these wind farms for the past 15 years, we can still see that there is stuff we don't know about how these communities are formed. And lastly, that there is an increase in a lot of invasive species, especially one that I thought was very obvious was Crepidula fornicata. And for me, I also worked on some samples from 2022 from Belwind, even though I didn't add it to my data. It was, yeah.
Questions? Well, start, yes. And thank you very much for your presentation. It's again very nicely illustrated. I would like to better understand what you meant by the community don't stabilize, and
That's what I want.
Yes.
what
Sorry, yeah.
is meant by that.
So I'll give an example of Belgium, for example. So what they noticed was that in the first 5 years, for example, after the wind farms were built, there was a sharp increase in the number of species. But then at the end, right now, if you go and collect samples, you'll notice that in the old wind farms at least, that there's only 4 species that are dominant. So there's no change. So there was a change in the abundance and the number of species present. So right now there's four major types are the Mytilus edulis, which is the mussel. There's Actiniaria and amphipods, especially Yasa and Corophium that like form around 90% of the biomass in the area. But originally in the first 5 years it increased, it shot up and then it stabilized to more or less these 4 species being most of them.
Okay. You mentioned several times that you have seen invasive species.
Yeah.
Could that compare to other sites that are not man-made, from man-made origin? Is it that those structures attract those invasive species, and then maybe you can tell me how they would have reached those areas? Or do you think that those species are there and you could find them in other natural structure?
So what happens is, so the species usually enter a region from shipping, for example, through ballast water, which already contain larvae. But originally, as I said, Belgium had a sandy bottom. So if these are hard substrate species, the larvae won't have any place to attach onto and settle. But now since we've added it, larvae suddenly had places to attach and settle, and then because they could outcompete the other native species, they grew in numbers.
Okay. Okay, yes, that's something that has been also described in other structures that might retain invasive species.
But all the ones that I spoke about have also been found to a certain extent in natural.
Okay, so how did you enjoy the speciation part? I mean, you talked about thousands of species that you had to analyze, so you would receive those buckets as you showed in the photos, and then under the microscope, and I'm sure it was a lot of time, but how?
Yes, it took me, I think, it took me around 1 to 2 weeks per sample to go from the bucket to having everything sorted and reserved in ethanol and counted. But I loved it, yeah.
Yes, thank you. Yes, good work. And again, yes, I can imagine how much work it must have been to be able to identify, quantify, and then.
Yeah, counting sometimes took a very long time, especially like YASA, for example, could go up to 10,000, 12,000. I had one with 19,000. I did subsample, but it still went down to 4,000-5,000 individuals that had to be counted.
To make the link maybe with the previous presentation, what would you expect as consequences now that you have an idea of what multi-communities are associated to those man-made structure? Do you foresee any consequences on other more organisms, fish and other? What's your idea on that? Can there be any subsequent effects, positive or negative?
I'm not sure about the organisms itself, but the wind farms themselves, for example, have a somewhat positive effect on the fish, for example, of the region, because they become a shelter. Because once you add wind farms, there's cables, there's actual wind farms, so bottom trawling has to be banned in that area. You can't fish, so the fish in there are healthier. There's more, I think, and if I am not mistaken, they are larger than the ones found outside of the ones that can be fished.
Okay. And would you recommend to include those concrete structure or biohut, as we mentioned, or any type of those, to use those poles as a way to attract and increase biodiversity?
So it's again the two-sided coin of it increases the biodiversity of the region, but naturally that biodiversity was never there. Right? It was soft substrate species that were present, and then we have added hard substrate species to.
Point. We sometimes try to add biodiversity, but maybe it was not there before.
Yeah.
It's a matter of what reference we use. Okay, thank you very much. Thank you.
Okay, thank you for a very interesting talk. And as mentioned, very relatable to what the previous talk. So I too was a bit confused by the term invasive species, but now it's clear, because when I thought invasive species, you mean something coming from Japan or something like that.
Crepidula is actually from.
They could.
Yeah, they come from everywhere. So there are the oyster, I think, is from the Atlantic.
But it's already found in the North Sea area, no? Or
yeah.
Yeah. So it's just a matter of a habitat that is now available. That
Yes.
Can,
Yes, therefore they act as the stepping stones. Where if it was found in only one small area of the North Sea, well, these have now spread everywhere. But it was just an example. Yeah.
Okay. So you said that you didn't include colonial species. Your analysis was based on abundance data, but why didn't you do presence-absence multivariate analysis, for example?
Because we thought that it would give us a better understanding of the smaller ones that we can, like we only take the countable ones, and then it gives us a better understanding of how many are there. So presence absence would just tell us this species is there. But for example, we have known for a long time that the non-native ones, for example, are there. But if you do abundance, it will give you a concrete idea of how many are there, or if there has been a drastic increase or decrease in the species.
Fair enough. Yeah. Okay. Good. Just one point, I think in your NDSs you should always show the stress level.
Yes, sorry.
That's a good point. So Belgium must be one of the countries in the world with the largest EEZ devoted to wind farms. And as you said, it's prohibited to do trawling and stuff like that. It's true that there is benefits, because over the years we've had many theses in IMBRC and EMBC on some of them on the fish communities that are associated with wind farms and using telemetry and showing really that they do aggregate fish and they do feed, they provide feeding grounds. But I'm just wondering, how can they continue? Is there some public, I mean, there are other stakeholders who are getting less and less access to the Belgian Sea, you know, recreational fishermen, for example. I mean, are there plans to build more and more?
As of now, as I showed you, there's only the plans to build. Yeah, these are the only planned wind farms, but that will, I think, become operational in 2030, if I'm not mistaken. But I'm not too sure.
With that, those two
Yeah.
are, I would say, like 25% of all it's a huge area that is devoted to wind farms. So, do you have any idea how much it contributes to the energy consumption of Belgium? Or
At the moment, I'm not too sure, but as I said, they want to make it the overall renewable to 17.5. Yeah, 17.5 by 2030. That's the target. But that's everything, onshore wind farms, solar.
Yeah. Okay. Well, as a comment, if you see that, when at the end, when your power bills they go down all the time because of the renewable energy, then renewables will be accepted much more easily than even though they may create limitations for other types of business. Okay, thank you very much. I have just a comment about you mentioned the stabilization of the communities. Okay, my point of view that in the marine realm stabilization, okay, there is this concept of the so-called climax communities is something that as a person I'm not too I don't believe that there are a lot in it that they consider, okay, this is the coast of Belgium. So I mean, it's the southern part of the Northern Sea. So it is an exposed sea. Yeah. And I assume that periodically there will be very strong,
it's a very dynamic.
And okay, consider when a really hard sea storm arrives, I mean, the strength of the waves will be huge. So probably many of these animals will be just ripped and taken off from the mills. And then that will create a new bare substratum. And then, depending on the time of the year when this happens, depending on the species that are reproducing at that time of the year, then their larvae will be around and they will settle there. So, let's okay. My point of view is that many benthic ecosystems in the marine realm should be seen really more like a mosaic, like patches in which you will have patches of old communities and patches of early, young, newly formed communities. So that's why that pattern. Okay, you mentioned this pattern in which apparently there was a decrease in the abundance. Now the other thing, okay, once again, look at asking from the point of view of a seaweed person, it looked like these surfaces were completely covered by the animal organisms. There was no seaweed at all.
Surprisingly, in my scrape samples, I didn't get any it was only animals, because I think maybe, I don't know, there's a lot of tube forming polychaetes and amphipods, and then it just gets covered by them and a canopy type situation is formed.
Okay, well, I guess if the sea bottom around is all sandy, probably not many sources of the seaweed.
Yeah, this is what it looks like.
It's a very different world.
Yeah.
Okay. If we have time for one quick question.
I mean, if I understood well, like now in that area where are the wind farms, you can fish like bottom shoreline, right? Are they thinking about also now sampling the soft bottom
The soft bottom is part of the, so I didn't mention it, but so my project was part of a larger project called WindMon, which is Belgium's wind farm monitoring project, which has been going on for 15-ish years. And they do, well, it's an impact assessment, so it does take into consideration everything. So there's other people doing, I think Kent University works with the Infauna and the soft sediment stuff.
Okay, good. Thank you very much. Okay, and now we have Nina that is going to talk about something which I am very interested in. So, Ethiopia, the Britain coast. Or is yours?
Do you know how many species are hidden in the sushi that you order in Japanese restaurant? Hello, my name is Nina Lopatina, and I would like to present you my thesis, Molecular Taxonomy and Distribution of Porphyra and Pyropia species along the Brittany coast. So Porphyra cinsullata is a very interesting object for aquaculture. It includes about 140 species totally, and in the South Asia it is used for a long time to aquaculture in industrial crops. However, there is no commercial cultivation and no control over the sexual reproduction in Europe on this species. So taxonomy, as I think, and the data of geographical distribution are very important data. And they should play a very important role in aquaculture because to choose the object for aquaculture, you should know, first of all, the species, the taxonomy of the species. And however, the taxonomy for this group of algae is not a very simple question. And there are many cryptic species inside. At least one molecular marker is needed for the species identification, according to the specialists who divided this genera Porphyra 13 years ago to 8 different genera. And some species could be difficult to separate even at molecular level. So if you see a morphology of Porphyra and Pyropia, you can see just very simple blades that represents very simple morphology, and but this is a different species. For example, these are two Pyropia species. This is Porphyra purpurea, this is Porphyra umbilicalis, Porphyra leuca, and Porphyra linearis in Roscoe. They consist of 1 or 2 layers of cells. Very simple, but difficult to understand the taxonomical, the border of the species and the volume of the genera of this group. So in 2020-2021, the research of algal genetic resources in Brittany was conducted, including Polyster project, which was has a goal to understand what we have in Brittany, like genetic resources. And research group who worked on this topic, they didn't. They had a problem with taxonomy of some barcodes that they obtained for Porphyra species. So the aim of my study, according to this, was first of all to try to resolve, if possible, the species complex between Porphyra linearis and Porphyra umbilicalis, abundant species in the area, using a new field sampling and DNA barcoding. And then I would like to try combining new data and polyester barcodes to refine the distribution and ecology of Porphyra and Pyropia along the Brittany coast. Here is shown the sampling map. We combine the polyester project study in 2019 to 2021, and this study from February to June that I did during my master thesis. So for each, we collected our algae on the shore, and we transformed them to the laboratory. For each collection, for each location, we tried to preserve some material in herbarium. We put some material on silica gel for the DNA extraction. We put some material on in petri dishes to culture, and we have a link, very strong link between all this material. So DNA barcoding is a very important tool nowadays to identify species, particularly algae species and red algae species. It's based on a COX-1 mitochondrial gene and RBCL plastid gene. These gene barcodes are well represented for Porphyra and Pyropia in a gene bank and in NCBI database, and they allow to delineate the species by phylogenetic analysis. So we extracted DNA using two protocols, nucleospin protocol and more simple, more shorter calyx protocol. And then we received extracted DNA. We amplified using PCR with specific primers. And then we visualize them into using electrophoresis, and not of them were amplified, unfortunately. Then we send them to Eurofans for sequences, and we obtained, this is the amount of the sequences that we obtained for now. Then we performed data analysis, which is phylogenetic analysis. And first of all, we should create a data set to understand our reference sequences. We took them from a gene bank. As a reference sequences, we try to use the sequences from type localities, which is important for taxonomy, and we edit our pool of sequences. We align them using MAF tool, then we editing them using gel view tool, and we our line sequence we use to construct our phylogenetic tree using RAXML tool in use Galaxy, and visualize them in Mega and ITOL. So our first result, we had this pool of sequences. It was 42 sequences in during polyester project, so that we received, not we, but previous studies received. And BLAST results showed very controversial results for RBCL and COX markers. And we constructed phylogenetic tree just for these sequences. As you can see, for RBCL marker, we can visualize some plates, but for COX marker, it's not possible because every sequence forms a very controversial clade. So then we edit our results, our pool of sequences then we received. And this, again, for RBCL, we can separate the species, but not for all of them. If the more sequences we edit, the more difficulties we dealt with to separate these two species. And for COX, even reference sequences, some of them were in the same clade. So my study has not been finished yet, but I would like to represent you a genetic material of I obtained genetic material for 3 species of. The most interesting for me is for Porphyra linearis, because briefly describe this life cycle. So Porphyra is generally is algae that has gametophytes and reduced filamental sporophytes. This was previously studied as Conchocelis species, distinct species. Then we was recognized that it is just the stage of the life cycle of this algae. And we collected some material from February in Roscoe. We put it on petri dishes, and then we collected some Conchocelis in April, but we didn't know that this is Conchocelis. We proposed that it is shells, and Conchocelis could grow on this. And we put it on aquarium. Then we sequenced this material and we received that this is all Porphyra linearis. Then it's not very easy to construct the tree for all 300, 400 sequences. I would like to present you just 3 for some of them, but it includes almost all the species that we found. The most abundant here is our complex. It's logical because it were our target species, Porphyra umbilicalis and linearis. But we can see also Porphyra dioica, Porphyra purpurea in 2 species of Pyropia. So for species of Pyropia that we found they are different genetically. They are and the different clades. We can separate them very simply, but on the morphological level, they are cryptic species. We can't separate them according to morphology. And then this is a map of distribution of Porphyra and Pyropia species in Brittany. The most abundant species, as we can see, Porphyra linearis, Porphyra umbilicalis, and Porphyra dioica. Porphyra purpurea we didn't find in the site. We just used polyester study, and it is generally in oyster farms. And Pyropia leucosticta, which is abundant in spring and early summer. Pyropia rosengurti, which is recognized now as Pyropia elongata, is a rare species. And we found from polyester sequences Neopyropia iezaiensis, which corresponds just in Cancale, where we have oyster farms, which corresponds to introduced species of algae. So these points I would like to bring into the discussion. 2 species of Porphyra linearis and umbilicalis form a species complex in genetic level, but occupy different ecological niche. Porphyra linearis is very small and grows in winter on the upper littoral zone, on splash zone. And the Porphyra umbilicalis is sessile and represents different morphology and grows on mussels, on shells, on other algae. They are so different morphologically, and what are the patterns driving these differences? Interrogression, hybridization, plasticity, I don't actually know. There are some supports in the articles that there is hybridization between these 2 species. It's like an idea in Iceland and other studies. So we observed the seasonal succession in Roscoe populations of Porphyra linearis and Porphyra dioica. In February, there is Porphyra linearis, according to our data, grown on between green algae, and at the same place on May, the end of May, it is Porphyra dioica. And then in other interesting results that we expected to find on the other side, Clider, just Porphyra linearis so upper, but we found two species together, and it was mostly Porphyra dioica. So on morphological, for now we are not possible to separate the species, but it supports by data of Varela Alvarez that describe from two locations the so-called winter form of Porphyra dioica. And then again, very interesting morphological similarity of two species of Pyropia. So my conclusion is preliminary conclusion that we recognize 7 species along the Brittany coast, 3 species of Porphyra, 2 species of Pyropia, and 1 apparently introduced species near Pyropia. Molecular taxonomy and phylogeny could help to understand the particularity of ecology and seasonal development of Porphyra. Only Porphyra linearis was known in Reskov in the upper littoral zone. But dioica replaced linearis in spring, and they can coexist in some populations. Pyropia species are less abundant, but different morphotypes of Pyropia leucosticta is often found in spring in the lower intertidal zone. Further research is needed on the population of Porphyra species in terms of genetic diversity and genetic differentiation. This material could be used for cultures. Thank you very much for your attention.
Thank you, Nina, for this nice presentation.
Okay, start ahead. Thank you, yes, for this presentation, and I learned a lot. I had no clue that there were so many species of Porphyra along the coast in Brittany. So how, I mean, this, you kind of started to explain that we didn't know when they would occur and not occur. Do you have any, yes, better sense on how we can first use these tools? What's the usefulness of having better understanding on how to use the molecular tool for the species identification? And also, how can you explain the co-occurrence of those species, if we have any explanation?
Yes. Molecular tools, it is barcoding.
Yes.
It is classical method. We can, it's,
what's the usefulness for this particular case to study Porphyra along different coasts?
It's useful generally, like you can use it in any case if you need to identify, because the cell genes are specific for red algae to identify species in the gene level, inside gene level. And normally,
so sorry, maybe my question isn't very clear, but so when you have done the analysis, is it from the algae or was it from seawater? Is it like environmental DNA?
Specific. It was
for the samples, right? Okay,
yes, it was this.
Okay,
it's a tissue of algae.
Okay. And can you use your result then for monitoring more with an eDNA approach if you have the sequencing that is now determined?
Yes, maybe yes.
Okay. Are all those species edible? Can we do not sushi, but with all of them? Is it something that is all the species?
Yes,
are consumable. Okay. Yeah, no, I can go ahead and then I will come back if I have any other question.
Thank you. This is very far from my area of expertise, but so these you have this complex of species. What about their geographic distribution? Do they all have the same geographic distribution or not?
Yes. Normally, yes, but I could say that I had just 3 months for all the work, and I should analyze more accurately this map and the data that I have for all the sampling. Because I just constructed the map, it was program which constructed, but then I should analyze which season they are represented and which normally they occupy different niches, so they could be together. It's not normal, but we know that Porphyra linearis it's a winter species mostly, and Porphyra umbilicalis could be all the year. There is no data before about seasonality of the species.
But in terms of latitude, beyond your study area, do they all have the same latitudinal range or not?
Yeah.
They do? Okay, so, and the idea is perhaps to start growing them to produce them on a commercial scale
yes,
in Britain?
Yes. But here in Jamaica, there was attempt to conduct the cultures of Porphyra dioica. But to know this, you should know life cycle, and it could be different for different species.
Mm-hmm.
It is important to first to have attempt in the lab, to have genetic material in the lab that we could say, this is Porphyra dioica. Yeah, we could give you, and you will reproduce. Or to have very simple tools, like I want to do it, but I don't know if I will be able to just to separate them morphologically, to find the characters that could separate them morphologically, that any person could say, okay, this is probably Porphyra dioica. If you need an expertise, you can give people who can expertise. But normally, in industrial scale, first of all, you should conduct the life cycle, you should finish it, and then try to reproduce it in cultures.
Presumably, we saw that there's like differences between them in the intertidal zone. So there must be differences in the effect of exposure.
Yeah.
So, and presumably there would be differences due to climate change eventually. So that would.
Yeah.
Are there any?
The cold impact, but Porphyra is a very adaptive. Yes, it's occupied a niche where other species could not live. It can be dry. For example, in this, as I shown, it was just no, it's not here. It lives here. It's 3 to 5 meters. It's like it could be dry, completely dry, the half of the day, and then so it's adapted to different changes.
To your
Okay.
expert.
Okay, now can you go to the slide of with the puzzle guy there?
Yeah,
inseparable. Okay, so look, let me understand exactly here. You mean that okay.
It's not understandable.
In sense, okay, you could not, okay, these sequences here,
Yes.
are something that, okay, why do you call them inseparable?
Because
is this actually
yes.
the phylogenetic tree in which, okay, see, you put only the linearis and umbilicalis?
Yes, I put just
okay.
two. I started with this.
Okay, okay.
Because
no,
in
well,
here,
simply this might be
market that it was in nature it will described as umbilicalis by collector, and then they suppose that will be umbilicalis. So it represents morphology of umbilicalis. And
okay, because the morphology of okay, fine. I understand. It's linearis. No, because I see that the bootstrap support. Okay, the support here is low, so I wonder if actually these things here might be actually a different species that here it ended up the close to linearis just because there were no other species. So okay, that might be the reason why okay you had the difficulties. Okay, not in sense that okay when you did the blast with these sequences here, do you remember which were the closest matches?
The first match umbilicalis and then some, 5 match linearis.
Okay.
It's for the many sequences. So yeah.
Maybe you discovered a new species. You should investigate more. But who knows? Okay.
I would like to continue.
No, but look at as the comment, actually what you found agrees perfectly with what I remember since the time I worked in Ireland. Was at the time, okay, in the area of Galway, it was exactly in the same linearis, mainly a winter species, and the umbilicalis, the more the other times of the year. Okay, no, I wondered the isolation of the conchosylis, on the skull of the shells. Is something that you did with the idea to try to develop some aquaculture. Are there any projects ongoing?
Okay. We collected near Porphyria linearis, and we expected that it could be the spores of Conchocelis there, and it was the case. We found them. Okay. And now it's preserved, so it can live. I will go, my practice will finish, but the material will stay in the school. And the material of this, and at least two additional species of Porphyria for using. And we know actually that this is Porphyria, this is Pyropia.
Of course. But it's very useful. I mean, this is information that is going to be very useful for the aquaculture as well. Because I remember, okay, this person, Elena Varela, that for which you cited in the paper, I know her, she used to work in the same laboratory in Ireland where I was in the early 2000s, and she was trying to develop also something like that for the aquaculture purposes. My understanding in this regard is that the species that we have here in Europe, from the point of the view of the taste, to make the nori, they are not do not taste as good as the Neopyropia yerzoensis, which is the one which is sold. Okay, we have a couple of minutes. We have time for another quick question. Does anybody?
Have you found any unexpected species, or was
no, just Pyropia, Neopyropia that it was introduced, but it just several times it was found in Kankari by the Osterpans ants, and we suppose that it is because of oysters brought from Japan. So we should have monitoring to the situation if the species will occupy more places or not.
Yes, if there is any competition with the other species, okay. Thank you.
Okay, thank you very much. Okay, very beautiful. Okay, so thanks very much and congratulations to the students who gave their presentations.